For our discussion in topic 12, we are going to be looking at really two different ideas, combining the two different ideas. So there we go. One about pathogens and how they have mechanisms in place to try to evade or trick the immune system so that they can survive. And then on the other side of the topic, we're going to be looking at how immunodeficiencies, not how they can arise genetically, but also just kind of an overview of the term immunodeficiency. We're not going to spend a whole lot of time on either of these topics or either of these ideas, and that's why we combine them together into topic 12. So the first part, the evasion or subversion of the immune system by pathogens. The reason we're not going to spend a whole lot of time talking about this is because of our focus in this class. So we really, taking human immunology as a course, we have the perspective of the human immune system, right? And we've been talking about how the immune system develops, how it learns, how it prepares itself, how it becomes activated, and how it fights infection. Now, thinking about the pathogen and how it tries to evade the immune system is actually taking the pathogen perspective. And I feel that that's better suited for a microbiology course. So if you do take microbiology with me, or you have taken microbiology with me, you might have, some of this might be review for you, or it will be preview for you, either way. And it's all about the perspective. And because we are focusing on the immune system and its work, we're not going to spend a whole lot of effort learning about the microbes and how they try to break that down, with some exceptions. We will talk about some big ideas. So here I start out with this perspective idea. So the human perspective, we have an immune system, and ideally it fights off any non-self foreign invader. And these tend to be called pathogens if they cause disease, if they damage tissue, or steal the body's resources, like a parasite. But in contrast, the other idea is that, hey, a pathogen doesn't want this to happen. And so taking that pathogen perspective and saying, hey, how can I trick or get away from the immune system so that I can survive and I can go on and grow and I can replicate? Now, the immune response to any pathogen, as we've seen in our talks, is that there's going to be a lot of complex molecular and cellular interactions between the pathogen and the host. We know that a lot of receptors have to meet up with their ligands, and there's signaling that occurs. And so where we have interaction between the pathogen and the immune system, that can be times or areas or points that can be targeted by the pathogen to try to interfere and end up surviving the immune response. So pretty much all pathogens in some way, shape, or form have evolved along with hosts, we call this coevolution, to reduce the effectiveness of the human immune response. So some of the big ways that pathogens tend to do this is by varying their genetic components. So changing up their genes, maybe looking a little bit different each time they infect a host. Maybe once they get into a host, they lay in like this dormant latency phase. They prevent phagocytosis of their cells. They can interfere with MHC molecules because we know how important they are for showing peptides to T cells. They can plain up just hide. There's a whole lot of other ways that that pathogens have evolved. We're not going to be able to cover all of them, nor would we want to in this course. We just want to give a brief overview. So, genetic variation. Streptococcus pneumoniae is the example that is shown in the diagram here, and it's one of the bacteria that have multiple serotypes. Salmonella has like 2,000 serotypes. I mean, there's just a lot of bacteria will have these different serotypes, and they're based on the different proteins that exist on their surface. And so even though the genus species is the same, the actual proteins that they express can be slightly different because they vary the genes that they will transcribe and translate. So even if somebody gets infected with, say, a strep pneumo, for example, at age 4, they can still become infected and have symptoms to a strep pneumo at age 7, probably because it's a different serotype. Even though it's the same genus species, it just looks slightly different. And so the antibodies or the memory cells won't recognize a different serotype. And so that's a really common thing that we see across a lot of bacteria. We can also talk about viruses too, and how genetics can be slightly different even though we're looking at the same virus. So influenza virus is the example that is on this page. Influenza virus, we know we need a new vaccine every single year because little pieces change, mutations occur, and those outer proteins can look different from season to season to season. And this is called antigenic drift. So we don't have serotypes when we talk about viruses, but we have different antigens on the surface. And even though the antigens can be pretty close, they're going to change just slightly. And those slight modifications can really trick the immune system into thinking that they've never seen this before, and they probably have never seen the exact combination of proteins. And so over time, then, when a virus accumulates enough changes, then it's going to look completely different to the immune system. And that's why we can get influenza multiple times in our life. Now, antigenic shift is when these changes happen abruptly. And so with antigenic drift, it's little slight mutations that occur every now and then throughout a couple seasons, and change eventually occurs to a noticeable point. But antigenic shift will happen whenever it wants. And usually this happens with influenza virus, particularly influenza A strains. And this can result in totally new hemagglutinin or neuraminidase proteins on the surface. And those are the proteins that interact with the host cells. So essentially, a brand new virus that the body and human population has never seen before can arise in an antigenic shift situation. Now, we had this happen in 2008. You may recall, maybe you know somebody that got sick, maybe you got sick, but in 2008, a strain of influenza called H1N1 changed. Well, it wasn't H1N1 at the time, and it was a combination of 2 different viruses, a human virus of influenza and then an avian strain of influenza. And pig cells are unique in that they have receptors for both avian influenza and human influenza. So a pig somewhere, they're thinking maybe Iowa, or somewhere in the middle part of the country, these 2 viruses both infected the same pig. There was recombination of the genes and ended up making a virus now that has avian genes in it and looks totally different to the human population. And this is what caused the pandemic of H1N1 or swine flu. Is probably something that you saw in your weekly reader or something come through, where the top picture just shows antigenic drift, where it slowly changes over season. Okay, so trypanosomes are super crazy. These are parasites. They are eukaryotic parasites, protozoans, and they cause the disease African sleeping sickness. And they regularly just change their proteins. It's crazy. So they infect somebody, they have a certain protein, the body responds to it, and they're like, oh, bring in the new protein, stop making that one, bring in a new one, and then they make the new one. And then the body's like, well, what the heck? Now this first organism isn't here, we got this new one. And so it makes the antibodies, a response to it, and then the trypanosome is like, oh, they figured us out, let's get a new one, stop making that protein. And so they can change their outer appearance and make the immune system super confused and end up causing quite lengthy symptoms. Crazy stuff. Okay, latency. This one we see with viral infections, where a virus can infect some cells. There may be some cytopathic effects, and there may be some symptoms that come about, but then it subsides. and it goes into a dormant or a latent phase, right? And when this is happening, not much happens to the cell that it's invaded. So in a normal situation, a CD8 positive T cell will recognize when a host cell is infected by a virus, and then it kills that one because it recognizes the peptides from the virus exposed on MHC class I molecules. And that's great, that's what we want to have happen. But when a virus such as herpes virus is a well-known one that causes latency or goes into a dormant phase, when they enter that quiescent state, they actually cause a reduction in the MHC class I molecules. And so they're not going to be replicating, they're not going to be generating new proteins, and so therefore there's not going to be this expression of these peptides to the cytotoxic T cells. The T cells are like, okay, well, we don't worry about it now. So this phase is called latency. It doesn't cause disease. However, you never rid your body of the virus. So herpes virus, like I said, classic example of a latency virus. Think about cold sores, for example. So herpes simplex 1, we have cold sores on like our nose and our lips and stuff, and at the nerve endings, because the virus lives back in the nerve ganglia. And then when the immune system is like stressed, or the lips get sunburned, or there's some sort of stress that happens either physically, or sometimes it can be mental, emotional stress too, can lead to a decrease in the immune system. And then that will reactivate the virus, because the virus is like, ha, 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 we can do it now, guys, let's go. And that would cause then an outbreak. And that's when you get a cold sore. So you often get a cold sore when what? You're super, super stressed out, you have an existing cold, or perhaps you go out and shovel snow and you didn't wear a face covering and your lips froze or got really, really cold. That's also can be a stressor on the cells. And so here's that nerve ganglia picture where it shows where the herpes virus resides, and then it will crawl down to the ends of those nerve tips, and that's where the sore will break out. And so we see that in the nose, along the lips kind of area. Then the viral replication in epithelial cells will produce process the peptides and then present them on MHC class I, and then CD8 T cells will find that out. But these herpes viruses are like, hey, I'm not going to go hang out in the epithelial cells. They make too much MHC class I, so they'll hang out in nerve cells because they don't have a lot of MHC class I, and they'll prevent the activation of CD8 positive T cells. So pretty sneaky, those herpes viruses. Okay, some bacteria cells, we know bacteria cells get gobbled up, right? Phagocytosis occurs because antibody is going to be an opsonine that attaches to the surface, or there is going to be complement deposited on the surface. However, some organisms are able to prevent the binding of antibodies, or if the antibody binds, they can produce a product that will block that FC portion and not allow for that receptor-mediated endocytosis to occur. So it actually like covers up the opsonins. So Mycobacterium tuberculosis, so that Staph aureus does that. Secretes this protein that prevents the binding of the FC portion. Mycobacterium tuberculosis actually just interferes with the phagocytosis pathway, or prevents that fusion of the phagosome with the lysosome, and then it can just start replicating inside of the cells, inside of those perfect little vesicles. Listeria monocytogenes is another bacteria that is able to, once it's engulfed, is able to escape that phagosome and grow in the cytosol. And so Listeria is actually an intercellular bacteria. And then there's just a whole bunch of other trickery. So I have a few examples here. The table that is from your text shows a whole bunch of other viruses that have the ability to do different things to a host. Toxoplasma gondii is a parasite This is the one that is associated with cat feces. And so if you're pregnant, you don't want to change the litter box because you don't want to inhale toxoplasma. And what it does is it prevents proteins from binding MHC, and therefore the T cell response isn't stimulated. Not a big deal for an adult, but it's a big deal for a developing baby. Treponema pallidum, schistosoma mansoni, they are going to evade antibody by taking human proteins and like making a cloak, an invisibility cloak essentially of human proteins, and making them look like their self. And so that will not stimulate then the antibody production or T cell responses. So whole bunch of different ways that pathogens have co-evolved with humans to try to escape immune system mechanisms. One thing that we need to mention is this idea of superantigens. So we know what an antigen is. It's going to be anything that can elicit an antibody response. So it's recognized, it can be self or non-self, but if it is something that can be recognized by an antibody. However, some bacteria, specifically like Staph aureus and Strep pyogenes, they are able to produce an exotoxin that actually is such a robust antigen that it will initiate or it will activate a whole bunch of T cell clones regardless of specificity. And that's why they're called superantigens, because essentially the specificity is gone, and if T cells are in the area of that toxin, they will become activated. Now, what do we know about T cells? We know they secrete a lot of cytokines because they have a whole bunch of helper functions. And when T cells start secreting, when all these different T cells get activated, they're all like, all right, time to go, let's secrete our cytokines. Well, heck, then we get a whole bunch of IL-2, we get a bunch of interferon gamma, we get a bunch of TNF alpha, all being released. And we know those are all pro-inflammatory cytokines. Now, all of a sudden, we have a whole bunch of inflammation occurring. But that's not all that happens. The superantigen can also cross-link MHC class II molecules on antigen-presenting cells, and then with the T cell receptor and the CD4 positive T cell. So it acts as like the co-stimulatory molecule. So it doesn't even make a T cell need that co-stimulation. It simply just says, nope, you're activated. See, it doesn't have that specificity. And that's why we get all of these cytokines going, all that inflammation, and that can lead to shock. That can lead to all sorts of systemic problems. So systemic inflammation, organ shutdown, all of this. So these superantigens can actually substitute for the specific antigen for a whole bunch of T cells. So let's look at that in a picture form. So in a normal, well, I don't have the normal situation here other than not having that superantigen, right? And so you have just the antigen presenting cell showing the peptide to the specific T cell, right? But when a superantigen comes in, it blocks that peptide. And so and then B7 and CD28 bind, that's a co-stimulation. So regardless of specificity, the T cells become activated. And then it is pure chaos. And this is what we call the cytokine storm. So perhaps you've heard of that in other papers, in other classes, in the news. But the cytokine storm is what results when we have overactivation of lots of T cells producing lots of inflammatory cytokines. So in summary, for part one, pathogens have really evolved a lot of different ways for just reducing the effectiveness of the immune system, including messing with the genetics through antigen variation, genetic shift, genetic drift. They can be latent and hide, or they can interfere with key elements of the immune system so that it just doesn't work properly. And these are all different ways that pathogens are able to survive the immune response and go on and cause infection. And that's why we don't have 100% immune response all the time, because pathogens evolve along with us. We make a change, they make a change. They make a change, we make a change. and we just co-evolve together. Okay, the second part is going to be very quick because the way this course is designed, you guys have been talking about these immunodeficiencies from day one, or maybe it was like day 4 or whatever, but these are our case studies. And so there are a lot of inherited immunodeficiency diseases, and we call these primary immunodeficiency diseases when they are inherited from mom or from dad or from mom and dad. And it, when you have an immunodeficiency, that renders an individual more susceptible to disease. Now, secondary immunodeficiencies are not due to genetic reasons, but rather to environmental factors. So sometimes drugs can be immunosuppressive because we can't have a lot of interferon gamma running around in some certain inflammatory diseases, and so we can purposely suppress the immune system, but they can lead to adverse effects. But then also we can have like secondary immunodeficiency diseases that are caused by pathogens as well, like HIV. So numerous inherited immunodeficiencies have been identified. I mean, we talk about a lot of them in class. There are hundreds and hundreds more. And each disease is due to a particular defect in a particular protein or glycoprotein, and that symptom then depends on the role that that protein has in the immune response. So there's over 200 primary immunodeficiencies out there that we know of for sure. The rate has definitely increased, or at least the numbers of them have increased because we've been able to do a lot more whole genome sequencing. However, like some of our case studies, most of them are very, very rare, and we don't see them but like 1 in a million or less, right? Primary immunodeficiencies, because they're genetic, they can be either dominant, they can be recessive, or they can be X-linked. And so that's no different than any other genetic disease that you've probably talked about in other science courses. And so dominant, that means that the defective allele can show up in children who inherit a normal functional allele from one parent and a defective allele from another parent, right? And so it needs to have only one copy. Where if it's a recessive allele, it's only going to show up if both parents pass on a defective allele. And then the parent who had that defective, well, both the parents that had the defective allele but not the disease are called carriers. So we can have heterozygous individuals then for a dominant trait, they'll get the disease, but for recessive trait, they will not have the disease, because for the recessive you need 2 copies of the defective, but the dominant you only need 1. You need to be homozygous recessive in order to have the recessive disease. X-linked is special in that the gene is actually attached to the X chromosome. And so we see this in individuals that have more than 1 X chromosome. Like if we talk about just binary male, female, XX for female, XY for male. A female is going to have less of a chance of actually having or presenting in a full immunodeficiency because they could have only 1 bad copy of that X and 1 good copy of that X. Whereas a male, if we're talking just regular binary male with XY, then that individual has a higher chance of having immunodeficiencies if it's X-linked because they only have the 1 X chromosome. So no different than any other inherited disease. So there are different types of inherited immunodeficiency disorders. You can have mutations in interferon gamma receptors, and that means that they're not able to respond to interferon gamma. That means macrophages won't be activated and you'll have decreased phagocytosis. You can have antibody deficiencies. If you have antibody deficiencies, you're going to have less opsonization, less classical, excuse me, pathway of complement activation, which will decrease the numbers of opsonins on the surface of bacteria, which will decrease the amount of phagocytosis that occurs. You can have defects in T cell health. This is a huge range of issues. If it's a CD4 positive T cell, then that is a bad news because that runs the whole show, right? But any of the T cells can have defective issues. Then that can affect antibody production, that can affect macrophage activation, or any of the other cells downstream of the immune response. You can have complement defects, you can have defects in phagocytosis, and you can have defects in T cell function. And these are all different ones that we have looked at specific case studies fitting into all these, with the exception of probably the interferon gamma receptor. I do not know that we looked at a case study that looked at that, but we have talked about interferon gamma. So, all in all, here's our big picture for topic 12. Deficiencies in the early component of the classical pathway then lead to a decrease in the production of opsonins, which will lead to the decrease of uptake of those pathogens by phagocytes. And so you'll have any organisms that are typically phagocytized, like bacteria. And so we see this leading to a lot of bacterial infections. I'm going to go to the left side now. The presence of antibody then, if we have an antibody issue, that is also going to decrease the amount of antibody that can serve as an opsonin. And so we end up having susceptibility to pyogenic bacteria. But the most severe, in the middle, would be immunodeficiencies that affect T cells, because T cells rule the immune system, and either directly or indirectly, through like helping other cells, will impair function. And so not only if you have impaired T cell function, you will also have impaired B cell function. You may also have impaired phagocytosis if there's not going to be a lot of macrophage activation. So when there is a T cell dysfunction that also affects the B cell dysfunction, we call these severe combined immunodeficiency disorders, or immunodeficiencies, sorry, or we call these SCID. And usually SCID is going to result from like HIV ends up causing SCID because it kills off the T cells, which will then decrease the effectiveness of the B cells. The other option would be the RAG enzymes. So if RAG1 and RAG2, which are vitally important for somatic recombination in both B cells and T cells, if the RAG enzymes are deficient, then we'll also end up with a SCID, or a severe combined immunodeficiency. So that's kind of the big idea. That, like I said, we didn't talk a lot about the immunodeficiencies because we've been doing that throughout the semester. With that, we'll see you guys in class.